We spent last week talking about descriptive statistics and how we use them to summarize a sample. This week, we will focus on inferential statistics and the elements of sampling probability that let us relate a sample to a population. The second major field of statistics is called inferential statistics. This set of techniques exists because it is often unfeasible to conduct a census. Because of this, we use inferential statistics to take a sample and make generalizable conclusions about a population. The two general uses of inferential statistics are hypothesis testing and estimating population parameters. We will focus mostly on the latter of these. We use inferential statistics to account for the differences that are known to occur because we are sampling a population instead of taking a census. Because we are sampling, our sample will not perfectly represent the population, and that is why we use inferential statistics to characterize this uncertainty. The thing to remember is that we start by trying to generate a representative sample of the population. We must meet the assumptions of sampling that our sample is unbiased and representative of the population. If we meet these assumptions, we can use the inference they afford us to draw conclusions about the population from our sample. Now let's talk a little more about the reasons we have to use inferential statistics when describing a population through sampling. We have two general ways of observing things in the natural environment. The first is direct observations, which is when we use some kind of instrument to directly assess the thing we are interested in. The second is when we use some kind of direct observation to infer something that is either too difficult, expensive, or just destructive to actually directly measure. Either of these approaches have several potential sources of error that impact their precision and accuracy, including selection bias, random errors, and systematic bias. We will look at how each of these contributes to our precision and accuracy. Before we look at how error enters our sampling design, we need to first understand the ideas of accuracy and precision. Accuracy is the closeness of our estimate to the true population value, while precision describes the agreement or variability between a set of measurements and is commonly characterized by the sample standard deviation. At the same time, we can describe error as the difference between a single observation and the true value. It is through our sampling design and data handling procedures that we're able to minimize the effect of sampling errors. It is important to remember when we describe a value as having an error of, say, plus or minus 5 centimeters, the word error does not mean a mistake. All we are describing is the precision in the measurement as a function of the experimental or sampling limitations. Moving forward, we need to distinguish between two ideas. Errors are small, unpredictable, yet constant things that affect the precision of a measurement. However, bias is a systematic effect that changes how we see objects in a population. The effect of selection bias is felt when a portion of a population is accidentally excluded from the sampling design, but we intend to actually include it in our inference. The only way to correct this issue is to collect more data from the excluded portion of the dataset. However, if we intentionally exclude a portion of a population we do not need to have inference for, there is no issue and we have not caused a selection error. We have simply redefined the bounds of our population. Often, when taking repeat measurements of a population, we have to deal with random errors that can result in small variations in our final results. These small errors are typically caused by issues in the measurement technique, such as not having a DBH tape perfectly level or at 4.5 feet every time we take the diameter of a tree. Another way we describe errors is as a systematic bias, which can occur when measurements are being affected by something that shifts all the measurements in one direction away from the true value. This is common with poorly calibrated instruments, but when caught, can be corrected for. It is unreasonable to believe we can eliminate all errors from our sampling designs. Mistakes are human errors that can occur at any time. It is easy to have these issues occur. Simple things like misreading a scale when in a hurry or putting a recording in the wrong cell of a datasheet can cause these simple errors. However, through training, practice, and diligence, as we carry out our work, we can try to minimize the occurrence of these errors. Error is inevitable when sampling. However, by collecting enough measurements, we can calculate what is best referred to as the most probable value. Generally, when we can assume we are dealing with a normally distributed population, the most probable value is usually the arithmetic mean. Conveniently, we can calculate the error or level of precision in a sample by subtracting the individual values from the sample mean. These differences are commonly called residuals, and we in turn use these to calculate our standard deviation.